In this video, we are going to have a quick recap about genetic code, general features of genetic code. The genetic code is written in linear form using the ribonucleotide bases that compose mRNA molecules as letters. Each word within the mRNA contains 3 ribonucleotide letters. Each group of 3 ribonucleotides, called a codon, specifies one amino acid. Thus, the code is a triplet. The code is unambiguous, meaning that each triplet specifies only a single amino acid. The code is degenerate, meaning that a given amino acid can be specified by more than one triplet codon. This is the case for 18 of the 20 amino acids. The different codons for a given amino acid are said to be synonymous. For example, UUU and UUC are synonyms for phenylalanine, whereas serine is encoded by the synonyms UCU, UCC, UCA, UCG, AGU, and AGC. Here we see a table which represents amino acids and their synonymous codons. We can read that from the figure, and after that, the code contains start and stop signals, certain triplets that are necessary to initiate and to terminate translation. Out of 64 codons, 3 do not code for any amino acid and called stop or termination codons. UEA, UAG, and UGA are stop codons. No internal punctuation, commas, is used in the code. Thus, the code is said to be commaless. Once translation of mRNA begins, the codons are read one after the other with no breaks within them or between them. There will be no commas or there won't be no breaks. The code is non-overlapping. After translation commences, any single ribonucleotide at a specific location within the mRNA is part of any one triplet. It is used to describe the genetic code as a universal code, meaning that the same code is used throughout all life forms. This is not strictly true. There are few examples of context dependent codons also. For example, UGA, a stop codon, also codes for 21st amino acid selenocysteine in some nuclei encoded mRNA. The code is nearly universal with only minor exceptions. A single coding dictionary is used by almost all viruses, prokaryotes, archaea, and eukaryotes. Some differences in the genetic code have been found, especially in mitochondria, chloroplast, some protozoans, and others. Here we could see some differences between the universal code and mitochondrial genetic code. Now we can see the coding dictionary. First position 5' end is represented on the left side margin, and third position 3' end on the right side margin, and second position on the top side. So we will be representing like this to get the complete codons. Next, a study question. If a hypothetical peptide has the sequence phenyltyrosine, methionine, proline, histamine, indicate why more than one nucleotide sequence is possible. Calculate the number of possible nucleotide sequence. So, indicate why more than one nucleotide sequence is possible due to degeneracy of codon. Then these systems have 2 codons. Proline has 4 and methionine has only 1. So we get 32 number of possible nucleotide sequence. In a heteropolymer experiment using 1/2C, 1/4A, 1/4G, how many different triplets will occur in the synthetic RNA molecule? How often will the most frequent triplet occur? There will be 3 raised to 3 or 27 triplets produced. The most frequent will be CCC, present 1 by 2 raised to 3 or 1 by 8 of the time. Next topic, Vobel hypothesis. Vobel in the anticodon. It was first proposed that a specific tRNA anticodon would exist for every codon. If that were the case, at least 61 different tRNAs, possibly with an additional 3 for the chain terminating codons, would be present. In 1966, Francis Crick devised the Vobel concept to explain these observations. It stated that the base at the 5' end of the anticodon also show non-standard base pairing with any of several bases located at the 3' end of a codon. So first base of anticodon and third base of codon is the Vobel position. For example, U at the Vobel position can pair with either adenine or guanine, while I can pair with U, C or A. However, the Vobel rules do not permit any single tRNA molecule to recognize 4 different codons. 3 codons can be recognized only when the usine occupies the first 5' position of the anticodon. Here we could see pairing combinations with the Vobel concept. G base in codon U or C, C with G, A with U, U with A or G, I with A, U or C. mRNA 5' end is there, 3' end is there, tRNA anticodon loop formation. There will be Vobel hypothesis happening. tRNA anticodon loop is there, mRNA 5', 3' and Vobel pairing between the anticodon on the tRNA and the codon on the mRNA. So that's all about these two topics of genetic code and Vobel hypothesis, anticodon pairing. So thank you for listening.